The dire wolf, a legendary predator of the Pleistocene epoch, vanished from the Earth roughly 13,000 years ago. Larger and more powerfully built than its modern grey wolf cousin, the dire wolf stalked the landscapes of North and South America, its haunting howl echoing through the millennia. Now, in a feat once confined to the realm of science fiction, scientists at Colossal Biosciences claim to have brought the dire wolf back from extinction. This remarkable announcement has sent shockwaves through the scientific community and ignited a fierce debate about the ethics and implications of de-extinction. Colossal Biosciences, a Texas-based company at the forefront of genetic engineering, has boldly ventured into uncharted territory, attempting to rewrite the story of life itself. The company's audacious project hinges on the revolutionary gene editing technology known as CRISPR. Using CRISPR, scientists can precisely target and modify DNA sequences within an organism's genome, essentially cutting and pasting genetic information with unprecedented accuracy. In this case, Colossal's researchers used CRISPR to edit the genome of a grey wolf, introducing genes from the extinct dire wolf. The result? 3 dire wolf pups named Romulus, Remus and Khaleesi were introduced to the world, marking a pivotal moment in the history of science and conservation. But the birth of these pups has also raised profound questions about what it truly means to bring a species back from the dead, and whether we should. Colossal Biosciences' approach to resurrecting the dire wolf relied heavily on the groundbreaking CRISPR-Cas9 technology. CRISPR, short for clustered regularly interspaced short palindromic repeats, acts as a kind of molecular scalpel, allowing scientists to make precise alterations to an organism's DNA. The process began by meticulously sequencing the dire wolf's genome from ancient DNA samples. By comparing the dire wolf genome to that of its closest living relative, the grey wolf, Colossal scientists identified key genes responsible for the dire wolf's unique physical characteristics, such as its larger size, powerful bite, and distinctive skull shape. Using CRISPR, the researchers then carefully edited the genome of a grey wolf embryo, inserting the targeted dire wolf genes. The modified embryo was then implanted into a surrogate mother, a domestic dog chosen for its compatibility and ability to carry the pups to term. This complex process, while groundbreaking in its potential, is not without its limitations. Critics argue that Colossal's approach, while resulting in animals that may resemble dire wolves, does not truly represent a resurrection of the extinct species. They point out that the genetic makeup of the pups is still predominantly that of a grey wolf, with only a small percentage of dire wolf genes. Furthermore, the complexities of gene expression and the interplay of countless genes in shaping an organism's traits mean that simply inserting a handful of dire wolf genes into a grey wolf genome may not fully replicate the extinct animal's unique biology and behavior. The debate over what constitutes true de-extinction is likely to continue as scientists grapple with the ethical and scientific implications of this emerging field. Colossal Biosciences' audacious venture into de-extinction has sparked immediate and widespread controversy. While the company heralds the birth of the dire wolf pups as a triumph for science and conservation, critics remain unconvinced, questioning the scientific validity, ethical implications, and potential environmental consequences of such an undertaking. At the heart of the debate lies the fundamental question of what constitutes true de-extinction. Critics argue that Colossal's approach, while resulting in animals that may superficially resemble dire wolves, does not represent a genuine resurrection of the extinct species. They point out that the genetic makeup of the pups remains predominantly that of a grey wolf, with only a small percentage of dire wolf genes. Moreover, the complexities of gene expression, the interplay of countless genes in shaping an organism's traits, and the influence of environmental factors mean that simply inserting a handful of dire wolf genes into a grey wolf genome may not fully replicate the extinct animal's unique biology, behavior, and ecological role. The pups, while genetically modified to express certain dire wolf characteristics, are essentially hybrids, a novel combination of genes never before seen in nature. Adding fuel to the fire is Colossal Biosciences' decision to announce the birth of the dire wolf pups via a media blitz rather than through peer-reviewed scientific publications. The company's reliance on press releases and high-profile media appearances, including a photo shoot with investor George R.R. Martin of Game of Thrones fame, has struck some scientists as a cynical attempt to garner publicity and attract investors rather than a genuine contribution to scientific discourse. The lack of transparency surrounding the project's methodology and data has further fueled skepticism within the scientific community. The de-extinction of the dire wolf has ignited a fierce debate about the future of conservation. Proponents, including Colossal Biosciences, argue that de-extinction technologies offer a powerful new tool in the fight against biodiversity loss, allowing us to right the wrongs of the past and restore ecological balance by bringing back vanished species. They envision a future where extinct animals once again roam the earth, filling ecological niches left vacant by their demise and contributing to the health and resilience of ecosystems. Furthermore, proponents point to the potential of de-extinction technologies to advance our understanding of genetics, evolution, and conservation biology. By studying the genomes of extinct animals and attempting to recreate them, scientists can gain insights into the genetic basis of unique adaptations, disease resistance, and other traits that could have conservation implications for both extinct and endangered species. However, critics counter that de-extinction efforts are a costly and potentially dangerous distraction from the urgent task of protecting the millions of species currently teetering on the brink of extinction. They argue that the vast sums of money poured into de-extinction projects could be better spent on traditional conservation efforts such as habitat preservation, anti-poaching measures, and combating climate change. Moreover, critics raise ethical concerns about the welfare of animals involved in de-extinction projects. The process of creating genetically modified animals, implanting embryos into surrogate mothers, and introducing these animals into the wild raises complex questions about animal welfare and the potential for unintended consequences. The long-term ecological impacts of introducing resurrected species into environments that have evolved in their absence are also unknown and potentially disruptive. The de-extinction debate highlights the profound ethical and ecological dilemmas we face in an era of unprecedented human impact on the planet. As we grapple with the consequences of our actions and strive to conserve Earth's dwindling biodiversity, the question of whether to bring back the dead will continue to spark both hope and controversy.